Hello, everyone, and welcome to Dana-Farber's Department of Data Science Frontiers in Biostatistics seminar. My name is Erica Feig, and I'm the program manager for the department. If you have a question for our speaker or the organizers, please use the Q&A tab or raise your hand to ask your question. Our faculty organizers will manage the Q&A session both during and after the talk. With that, I will turn it over to Nabia Teo to introduce our speaker for today.
Thanks, Erica. So today we are very glad to welcome Dr. Emily Zabor, who is an assistant staff biostatistician at Cleveland Clinic in the Department of Quantitative Health Sciences, with a joint appointment in the Taussig Cancer Institute. She also holds an academic appointment as an assistant professor of medicine at the Cleveland Clinic Lerner College of Medicine of Case Western Reserve University. Her research interests are in early phase clinical trial design and statistical methods for observational studies with applications to oncology, and her expertise is in applied statistical analyses. The commonly used methods in her work include survival analysis, longitudinal analysis, prediction modeling, and methods for epidemiologic research. And while she primarily works in cancer, she's also interested in applications to international health, public policy, and social sciences. As a lot of us, I think, work in R, is what's interesting is she also programs in R and has an interest in educational activities involving R. And she has a master's degree in biostatistics from the University of Minnesota and her PhD in biostatistics from Columbia University. Today, we welcome Emily to discuss her work in early phase clinical oncology trial design. And so thank you so much, Emily. We're looking forward to your talk today.
Thank you so much for the introduction and for inviting me to speak today. As they mentioned, please feel free to drop questions into the Q&A at any time, and I'll try and keep an eye on it or get interrupted to answer those as I go. I'm happy to take questions. So I'll be talking today about some of the methods development I've done in early phase oncology clinical trials, and specifically focusing on the case study of Atezolizumab development to motivate this work and to describe the results of some of the studies I've been doing. So I'm sure a lot of you are familiar with oncology trial designs, but I'll just give a little background to make sure we're all on the same page about what kind of the problems are that I've been thinking about. Early phase oncology trials have been changing in response to the changing treatment landscape in oncology. So traditionally, development of drugs in oncology was focused on cytotoxic chemotherapies, where the toxicity increased with the dose and the efficacy also increased with the dose and the toxicity. And so small phase 1 studies would be conducted, usually using some type of 3+3 design or model-based design to identify the maximum tolerated dose in that setting. But with molecularly targeted agents and immunotherapies being the focus of drug development in more recent decade or so, the maximum tolerated dose does not always exist or is not always reached in these trials. And so a lot of times what's happening is dose expansion cohorts are being added in an ad hoc manner. So the sample sizes are getting larger and larger. And these trials are becoming a little unwieldy. And so some new methods are needed to address the specific treatment setting. And one area of particularly rapid development in oncology has been in immune checkpoint inhibitors. And so this is just kind of showing a timeline, not to the present day, but up through 2018 of immune checkpoint inhibitor development, with the first being approved in 2011, which was ipilimumab for melanoma, and then kind of a number of years of break, and then an explosion in these immune checkpoint inhibitor development, really rapid pace of development in this area. And one of those drugs was atezolizumab, which was approved for a number of different indications. And one of them was metastatic urothelial carcinoma in May of 2016. And that's the context I'm going to focus on today. So atezolizumab is a PD-L1 checkpoint inhibitor. And what that means is that it binds to the PD-L1 protein on the tumor cell, and it blocks the binding of PD-1 on the immune cell to PD-L1 on the tumor cell, allowing the immune response to initiate tumor cell death. And it was studied in phase 1 with a kind of a traditional 3+3 design to start, going through these doses that you can see here. These were the doses that were of interest to start in all comers. And actually the MTD was not identified and was not reached in this study. And so a number of phase dose expansion cohorts were initiated, and the pre-planned ones were in non-small cell lung cancer, renal cell carcinoma, and melanoma. And then there was an option for some other tumor types in the expansion cohorts. And you can see on this schema that they looked at a number of different dose levels since the maximum tolerated dose hadn't been reached in the dose escalation phase. And so in the urothelial carcinoma setting, there were 95 patients enrolled in a dose expansion cohort for this indication. And they found an overall objective response rate of 40% in the biomarker subgroup of interest, which was this IC23 subgroup, which has to do with the percentage of immune cells for PD-L1. And so a phase 2 study was done in this setting based on those results, which were considered promising. And they found in that biomarker subgroup of interest, again, a 26% response rate. And that was used as the basis for accelerated approval in this indication. So in 2016, atezolizumab was approved for the treatment of advanced urothelial carcinoma. And after that approval, the phase 3 trial was ongoing, and eventually those results came out, and the results of the phase 3 trial indicated that there was no difference between atezolizumab and standard of care chemotherapy. So these results I'm showing here were just for the biomarker subgroup of interest. Actually, over 900 patients were enrolled into this trial across all subgroups. But in the biomarker subgroup of interest, patients were there was a hazard ratio of 0.87 for the comparison of atezolizumab to chemotherapy, and that was for overall survival, and that was the primary endpoint. So if we look at the overall response rates in this subgroup, we see that the overall response rate in the atezolizumab arm is not that different from the phase 2 results. We see 23% here, and in phase 2 it had been 26%, but the overall response rate in the chemotherapy group is also very high at 21.6%. And so this was a failed phase 3 trial. After those results came out, the indication for atezolizumab in bladder cancer was eventually withdrawn by the sponsor in March of 2021, though that was a number of years after those phase 3 results were published. So that was kind of the development path of this drug and this indication. And it leaves something to be desired with the design of these trials and the way that they were implemented. And so I'm going to propose some ideas for some different ways that trials like this can be conducted to avoid some of these pitfalls. Of course, with the caveat that, hindsight is 20/20, so it's easy for me to say this now, looking back on the results of these trials. So the first area I'll talk about is dose expansion cohort design, which is related to the phase 1 part of this study. So the use of dose expansion cohorts in phase 1 oncology trials is increasing over time, which I mentioned back in the introduction, related to the kind of changing treatment landscape in oncology. And this isn't completely up to date, but in this meta-analysis by Manji et al. That was published in 2013, you can see that the use of expansion cohorts in phase 1 trials increased from 12% in 2006 to 38% in 2011, and I would imagine it's probably only increased further since then. And in a separate meta-analysis by Bugano et al. In 2017, they found that trials with expansion cohorts had a higher rate of phase 2 success, but that larger trials were not necessarily better. So in this plot from that paper, we have the number of patients in the phase 1 expansion cohort on the x-axis and the probability of at least one positive phase 2 trial result on the y-axis. And we see an increase in the very beginning up to maybe around 15 or a little less than 20 patients, and then kind of a plateau in the probability of having a successful phase 2 trial, with the number of patients increasing after that. So my goal here was to develop a pre-specified dose expansion design that can maintain traditional frequentist error rate control while also making efficient use of limited resources, meaning trying to keep the sample size small. So one tool that I am going to use for this is futility stopping, which can be used to achieve multiple phase 1 expansion cohort goals by stopping the trial early for futility. It maintains ethics for participants and also can be an efficient use of resources by keeping the sample size small if a drug really isn't working. And it's still possible to control the type 1 error and power with the inclusion of futility stopping. So here I'm going to consider the setting of a one-arm clinical trial that has a binary response endpoint. So each patient either has the outcome so that their X value is 1, or they don't have the outcome, so it's 0. And then we are interested in the phase 1 setting, usually in testing the null hypothesis that our response rate is less than or equal to some null response rate versus the alternative that our response rate is greater than or equal to some alternative response rate. And so I'm going to be using the Bayesian statistical paradigm for these methods throughout my talk, which has been the basis for sequential statistical learning and has been proposed quite a bit for use with futility monitoring in early phase trials. So this is not a new idea here, but we have our prior, which is going to be a beta prior, and specifically for all the results I'm showing today, I'm using a beta 0.5 prior. And with our binary endpoint, we have our binomial likelihood. And so when we combine those two, which are conjugate, we have our beta posterior distribution. And then we would declare our treatment to be efficacious at a specific posterior threshold theta if our probability of the response rate exceeding that null rate, given our data, is greater than that posterior threshold theta. And so predictive probability monitoring is going to be employed here to stop the trial according to a threshold, a futility threshold. And so the posterior predictive distribution of the number of future responses in the remaining patients who are yet to be enrolled follows a beta binomial distribution with this form. And then our posterior predictive probability can be used to stop the trial, which we would stop for futility at our predictive threshold theta star if our posterior predictive probability falls below that threshold. So the challenging thing in using these methods is selecting the thresholds. And so simulation-based methods will be used to evaluate a grid of thresholds to compare the operating characteristics here and to identify a feasible trial to implement. So in the simulation results I'm going to show, I'm looking at a threshold of 10 different posterior thresholds ranging from 0.9 to 0.99 and 4 different predictive thresholds, 0.05, 0.1, 0.15, and 0.2. And so this grid is a combination of 40 different potential designs, which will have their operating characteristics simulated to calibrate these thresholds for our ideal design. And so the new work that is here is I'm proposing 2 optimization criteria to calibrate these thresholds. And I'll describe them visually first, and then I have some more details on the next slide. But on the left here, the first criteria I'm calling the optimal accuracy design. And this criteria has to do with the type 1 error and power. So each of these points on this plot represents one of the 4D design options that I showed previously on the slide. So each point is a design based on a different posterior and predictive threshold. And on the x-axis is the type 1 error associated with that design based on simulation results. And on the y-axis is the power. And so then the optimal accuracy design can be selected as the design that's closest to the top left corner of this plot. And I'll mention some options for weighting on the next slide. And then the second criteria is the optimal efficiency design, which again, each of these points represents one of the 4D potential designs based on different combinations of posterior predictive threshold. But now the x-axis is the average sample size under the null hypothesis for that design. And the y-axis is the average sample size under the alternative for that design. And again, we can identify the point that's closest to the top left corner of this plot is the optimal efficiency design, with the caveat that there needs to be some upfront filtering of these designs to constrain the type 1 error and power. Otherwise, it's possible to select an optimal efficiency design that has much too low type 1 error or too low power. So these criteria are defined using Euclidean distance to that upper left point on both of those plots, with the option of weighting based on the clinical context. So if you had a reason for either the type 1 error or power being more important, you could supply weights to these distance measures. And similarly, if there was a clinical reason for either the sample size under the null or the sample size under the alternative being more important, you could similarly put weights onto the Euclidean distance. And so going back to the Atezolizumab study, the expansion cohort, the dose expansion cohort for metastatic urothelial carcinoma wasn't even included as part of the original trial design. As I mentioned earlier, the 3 originally planned expansion cohorts were non-small cell lung cancer, renal cell carcinoma, and melanoma, with the option of these other tumor types. So this was added through a protocol amendment. However, the protocol amendment did not specify a design for this expansion cohort in metastatic bladder cancer. But the original 3 expansion cohorts were planned to have a total sample size of 40, with a single interim futility look to stop the trial if there were 0 responses in the first 14 patients, so a single look for futility. And the protocol stated that this futility rule was associated with a 4.4% chance of stopping if the true response rate was greater than or equal to 20%. But the metastatic urothelial carcinoma in practice enrolled 95 patients, not 40. So it's a little unclear what the actual design of this study was, or if there was a specific statistical design to this 95 patient dose expansion cohort. So I designed a simulation study based on my best guess at what the original trial design was under a variety of potential combinations of some of these tidbits of information. So I looked at 2 different null response rates, 0.1 and 0.2, and 2 different alternative response rates, 0.2 and 0.3. And then I looked at both the sample size of 40, which was the planned sample size for the other dose expansion cohorts in the protocol, and the sample size of 95, which was the actual sample size used in the trial. And I'm going to be comparing my proposed optimization criteria with the original trial design that had a single futility look. So I'm showing the results here just for the optimal efficiency results, and this plot is only for the simulation setting of a sample size of 95. So the panels are for the different combinations of null and alternative response rate. So null of 0.1, alternative of 0.2, null of 0.1, and alternative of 0.3 in the middle. And on the right is the null of 0.2 and alternative of 0.3. And so each of these panels has the average sample size under the null on the x-axis and the average sample size under the alternative on the y-axis. And the dotted points are one of the sequential predictive probability points, with the orange point indicating the optimal efficiency design. So that's the closest point to the top left corner, and no weighting was used in this simulation. Then the triangle represents the original protocol design with the single futility look. And so we see that in all cases, the optimal efficiency design has lower average sample sizes under both the null and the alternative as compared to the protocol design. And to see the numerical results here, we see that the optimal efficiency design also has higher power, and that the protocol design, which is the bottom row in each of these sections of the table, has much too low type 1 error and also too low power. The type 1 error is below 0.1 in all 3 combinations of null and alternative, and the power is just around 50%. And they also have higher sample sizes. So across all of these different measures of type 1 error, power, and the sample sizes, both the optimal efficiency and the optimal accuracy designs do better, but the optimal efficiency design has even better characteristics than the optimal accuracy design in this case. So one of the challenges of designs like this is that they are computationally intensive. As I mentioned earlier, you have to do a simulated grid search through the 40 possible or however many, different threshold combinations you're considering. In this simulation, I looked at 40 combinations and to get the operating characteristics and select a design. But the nice thing is that can all be done at the design stage, and then you can obtain the decision rules for the selected design up front so that there's no need for inter-trial calculations at all. So the decision rules can be either mapped to a table, or this is a plot of the decision rules where across the x-axis we have our sample size at interim analysis. And I think I forgot to mention that this trial was in my simulation study, I was monitoring after every 5 patients to allow for early stopping after potentially every 5 patients were enrolled up to the total of 95. And so here you could just make a decision at any point in the trial based on the sample size at the interim analysis and the number of responses, whether you would proceed, which is a green box, or stop the trial early if you fell into the red area on this plot. So that was dose expansion cohort design. And now I'll move on to discuss some similar, yeah.
Sorry, I had a question about that.
Yes,
just from more
please.
of a kind of a practical aspect.
Yes.
Obviously, stopping every 5 patients would give you the opportunity to stop your trial quite early if it's no signal. Just from a practical aspect, though, sometimes it can take a while to get your responses, right? Yes. So to wait for each, like for the 5, so how is this practically implemented? Like, is it that you need to see all the to have assessed the responses in all the patients and then you keep going, or doesn't that make the trial quite long?
Yes, so that's a really good question. So if you're in a setting where the toxicities are observed very late, it would not be practical. But I mean, in a trial like that, you do still need some sort of interim monitoring for both futility and toxicity. But I mean, if your responses are observed quite late, in my experience designing trials, most of the time responses they're looking in a relatively short window. And so designing something like this would be feasible. And with the immunotherapies, we're more in a setting where responses can be seen relatively quickly, but toxicities might be seen quite late. So it's more of a problem on the toxicity monitoring side of things. But yeah, it might not be practical depending on the setting. And if you have continuous enrollment and you haven't seen the responses for the preceding patients, you could end up in a situation where you do have quite a few patients enrolled, and then all of a sudden you're stopping for futility once responses are coming in. So no, that's a challenge. Does that answer your question, kind of?
Yes, it did. Yes, thank you very much.
Okay, so just a little reminder about these results from the phase 2 trial in the atezolizumab metastatic bladder cancer setting. This top box up here in the top left is the phase 2 results. So in the biomarker subgroup of interest, they saw an overall response rate of 26%, which they considered promising. And this trial was designed in comparison to a historical control rate of 10%. And then down here, we're looking at the phase 3 results for the biomarker subgroup of interest, in which the treatment group had an overall response rate of 23%. So not that dissimilar from the phase 2 result, but the chemotherapy group had a overall response rate of 21.6%, which was much higher than the historical control rate of 10% that had been used to plan the phase 2 trial. And part of the problem can come into a trial like this because these historical control rates that are being used to plan trials arise as population averages, so they may not apply to a biomarker targeted subgroup like this. And as I mentioned earlier, this atezolizumab phase 3 trial results came out in 2018. But the drug had already been approved with accelerated approval in 2016, and still for 3 more years after that phase 3 trial came out, showing no improvement in overall survival for the treatment, the the approval was out there on the market. And this is what's known as a dangling accelerated approval, where the accelerated approval is out there, but there has already been a phase 3 trial showing no improvement in the primary results. And interestingly, there were actually 10 such dangling accelerated approvals among anti-PD-L1 antibodies alone, just between 2015 and 2021. And here's a abbreviated table of some of those. And some of them are still have their accelerated approvals on the market despite negative phase 3 trial results. So this is kind of a problem that's coming up quite a bit, that these drugs are being approved on the basis of potentially poorly planned phase 2 trials, and then that approval staying out there for some time, even after definitive trials have shown no improvement in results. So my goal here was to develop a randomized phase 2 trial design specifically for biomarker targeted subgroups using the optimal efficiency criteria that I just mentioned in the context of phase 1. So now I'm going to consider the setting of a randomized 2-arm trial that has 3 biomarker subgroups. So this is going to be closely based on the setting of the Atezolizumab study. So the subgroups have equal population prevalence of 33%, with a null response rate based on the historical control rate used in that trial of 10%. And then subgroup specific alternative response rates of 10, 20, and 30%. In the IC0, which is no PD-L1 immune cells, IC1, which is less than 5% PD-L1 immune cells, and the IC2-3 subgroup, which is the subgroup of interest. So in this case, the biomarker is hypothesized to be predictive, though, as we just saw from the actual results of the trial, we do know now that the biomarker is much more likely a prognostic biomarker, where the response to both the chemotherapy and the atezolizumab treatments were much higher in the biomarker targeted subgroup than in any of the other subgroups. But I'm still basing this design on the hypothesis that the biomarker was predictive, which was the original hypothesis of that study. And I'll come back to this point in a little bit. So I proposed 3 different randomized trial designs, and I'll go through each of them and then show some simulation results of how these all performed. So the first I'm calling the pooled control arm design. And in this design, we enroll all patients and then randomize them to either the chemotherapy or treatment arm in a 1-to-3 ratio. And I'm basing that on the distribution of those treatment, on the fact that I have these 3 biomarker subgroups. So then in the treatment group only, the biomarker testing is conducted and patients are stratified into their 3 biomarker subgroups. And then at the end of the trial, we would compare each of these subgroups back to the pooled control arm. The second proposed design I'm calling a stratified control arm design. So again, we enroll all patients, but then we conduct the biomarker testing on all of them, stratify them into the 3 biomarker subgroups of interest, and then within each biomarker subgroup of interest, randomization is conducted in a 1-to-1 ratio to either the standard of care or the treatment arm. And so then at the end of the trial, each treatment arm is compared to each standard of care arm within each biomarker subgroup. And the third and last design I'm calling the enrichment design. And in this design, the stage 1 of this design is the same as the pooled control arm design. And at the end of stage 1, we would select a winning stage 1 subgroup. And this would be based on the subgroup that has the highest posterior predictive probability, subject to some lower bound. And I'm proposing that in this simulation study, I used the lower bound of 80, the 80th percentile of the maximum posterior predictive probability to target a 20% type 1 error rate of selecting a subgroup to carry forward to stage 2 if actually none of these subgroups was promising. So then in stage 2, you enroll all patients and conduct the biomarker testing, and then you retain only the patients who are part of the winning stage 1 subgroup, and then randomize those patients in a 1-to-1 ratio to either the standard of care or the treatment. And then we will pool the stage 1 winning subgroup patients with our treated patients and compare them to this chemotherapy arm or to the standard of care arm. So those are the 3 design proposals, and here are some simulation results where I'm showing each of the panels now represents one of the randomized designs. So the one on the left is the pooled control arm design, the middle is the stratified control arm design, and the right is the enrichment design. And these are the results for optimal efficiency, where we have the average sample size under the null on the x-axis and the average sample size under the alternative on the y-axis. So, and I investigated the same posterior and predictive thresholds that I showed in the phase 1 setting, so the 40 different designs. So you can see here that all designs result in much lower average sample sizes than the utilized phase 2 sample size of 310 patients, which is what was the actual realized sample size of the Atezolizumab phase 2 study. This orange point represents the optimal efficiency design on each plot. And I'll show some numerical results as well, which indicate that all 3 designs lead to reasonable type 1 error and power. For the pooled and stratified designs, we only have subtype specific operating characteristics, but you can see for our subtype of interest in both of these designs, we have a type 1 error of 7% and a power right around 80%. So those are good properties. And in the enrichment design, we only have an overall operating characteristics. So we see in the stage 1, we have a 9% type 1 error and a little higher than 70% power. And similarly in stage 2, we have a 9% type 1 error and a power greater than 80%, again, 86% here. So these are reasonable design characteristics that I think would be acceptable to most investigators. Oops, sorry. So I'm coming back to this problem of a predictive versus a prognostic biomarker and how to even choose between these 3 designs that I proposed. And it would really depend on the clinical setting and also the financial setting of the trial. So just some pros and cons of each of these designs. The pooled design has the smallest sample size and also does the least biomarker testing. So if your biomarker testing is either very expensive or very invasive, the pooled control arm design could be a good option for that reason. For the context of a predictive biomarker, any of these 3 designs will be able to identify that for you. For the context of a prognostic biomarker, I'm not sure that any of them really can. Maybe the stratified design would still fail because you would be directly comparing, a treated group to an untreated group, and they would probably both do well in the context of a prognostic biomarker. But at least in this case, you would get that information and you would find that out before you proceed to phase 3. So my conclusion from this is that the stratified control arm design would have been the most, the best choice in this setting of the Atezolizumab trial. It averages 214 patients, a sample size of 214 patients under the alternative, as compared to the 310 that were actually used in the phase 2 trial. And the average sample size is even smaller under the null. But in addition to that, you would find out the information by using the control arm within each of the biomarker subgroups. You would find out the information that you're probably dealing with a prognostic biomarker and that there was no difference in response rates between the 2 groups. And so you would also avoid enrolling the 935 patients who were studied in the phase 3 trial based on the incomplete phase 2 results that were based on a historical control rate that wasn't accurate for the targeted subgroup of interest in this trial. Any questions about that phase 2 methodology before I move on to my last little segment? Oh yeah, and I just was also going to mention that I do have an R package that implements all of this methodology. It's called PPSeq. It's available on CRAN and also on my GitHub page, which you can see here. And it has a pretty detailed website that has some vignettes and helpful information to get started using it for anyone who did want to try and design this. I think it's fairly user-friendly. I think one of the problems with a lot of these new trial designs is the methods sound great, but they're not that easy to use. And so I was trying to make this something that would be able to be implemented in practice by anyone who was interested in it. All right, so the last topic I'm going to talk about is basket trial design, and this will be the briefest of the 3 segments. So conventional basket trials are trials in which we have multiple different tumor locations, tumor types. So you might have breast cancer and gastric cancer and lung cancer as your different tumor types, but they all share a common genetic or biologic target of interest that the treatment, that's a single treatment that is being used in the basket trial, is targeting. And these trials are commonly conducted in the phase 2 setting, and often with the goal of trying to get approval of a treatment in a tumor agnostic setting. So the Atezolizumab dose expansion study really was a basket study with these different, this is a different picture of this, excuse me, study schema where we see not only the 3 originally planned dose expansion cohorts of melanoma, non-small cell lung cancer, and renal cell carcinoma, but also some of the other cancer types that were added through protocol amendments, including the urothelial bladder cancer and also small cell lung cancer, triple negative breast cancer, ovarian cancer, and glioblastoma were also added through protocol amendments. So this was a basket trial, but in the setting of atezolizumab, the expansion cohorts were designed and studied independently, which is a somewhat common approach to basket studies still. So my goal here was to incorporate control for multiple testing into a basket trial design that shares information across baskets to leverage the strength of the multiple subgroups design. Because one of the challenges of a basket trial with this number of different subgroups is that, first of all, you're increasing your risk of false discovery by doing multiple independent tests within each of these baskets. And second of all, depending on the context, you might have some of these cancers that are more rare than others, or you have trouble enrolling in one of these baskets versus some of the others. So you might end up with differential sample sizes across baskets that might make it easier to make a strong conclusion in one and harder in some other cancer type. So these are some of the issues of independent basket studies that I was hoping to address. So the basis of what I'm proposing is the multi-source exchangeability model, which is methodology basket trial design that basically it considers all pairwise exchangeability relationships between each pair of baskets. So in this little schema, can you still hear me okay? I just got a little pop-up saying my internet connection was unstable.
Yeah, you dropped off for a little bit, but you've picked up again on my end.
Okay, great. So in this little schema here, where we have 3 baskets indicated by these 3 Ys, this is just showing that, 2 of the possible pairwise exchangeability relationships would be if Y1 and Y2 were exchangeable, but Y3 was separate, or maybe Y1 and Y3 are exchangeable and Y2 is separate, and so on and so forth. And what the methodology does is it estimates all of those possible pairwise exchangeability configurations and identifies the one that has the maximum posterior probability. So going back to the atezolizumab study, in the dose expansion study, they actually ultimately studied 11 different cancer types. And they're all listed here in the order from the one with the highest overall response rate to the lowest. And what we can see here is not only are there vastly different response rates from 30% in melanoma down to 4% in prostate, but also there were very different sample sizes from a maximum of 112 in the triple negative breast cancer cohort down to just 9 patients in ovarian. And so what we really want is to use these Bayesian methods where we're sharing information across cohorts that have similarities so that we can leverage the overall sample size that's available across these 11 cancer types. And these are just the references for the response rates on this slide for full, to attribute those properly. So one of the ways of seeing the relationships that come out of the multi-source exchangeability model is a network graph. And so what I'm showing here, each of the nodes is one of the 11 cancer types that was studied in a dose expansion cohort. And this is not a simulation study. This is a reanalysis of the actual data, the actual response rates from that study. So we can easily see that there are kind of three clusters of cancers here. And what's in this network graph is the color of the node represents the posterior probability of exceeding the null response rate. So this yellow color is a high posterior probability, and this dark purple color is a low posterior probability. And then the edge weights connecting each basket, the thickness of the edge weight represents the posterior exchangeability probability between a pair of baskets. So the thicker the line, the higher the posterior exchangeability probability between any given pair of cancer types. And I've only plotted the edges that were at least were greater than 0.25. So that's why not every edge is included here. So we can see we have here a cluster of a few cancer types that have a high posterior probability and are very exchangeable, a couple in the middle that kind of have a middle posterior probability with high exchangeability, and then these ones over here with very low posterior probability that are also highly exchangeable with each other. So my proposal here was to incorporate false discovery rate control with the multi-source exchangeability model. And I was interested in false discovery rate control since it aligns with the discovery-oriented objective of early phase basket trials, where we really do want to identify cancer types where the treatment is promising to carry on to further study and are willing to accept maybe a higher family-wise error rate as a trade-off. So the way this was implemented is to obtain the posterior probability that each basket, little j, exceeds the null response rate, to order those posterior probabilities from the largest to the smallest. And then for a given threshold of posterior probability, which I'm denoting phi, identify the largest k such that the basket with the that posterior probability is greater than k over capital J, which is the total number of baskets times that threshold phi. And then we declare all the baskets that have a posterior probability greater of k or higher to be significant at that threshold phi. So looking at this, going back to our 11 cancer types in atezolizumab, and here I'm just looking at a posterior threshold of 0.95 as an example. And now I have these baskets ordered in order of their posterior probability of exceeding that null response rate. We see that using a threshold of 0.95, we would have declared 7 of these baskets to be promising at that rate, and these 4 would have been declared not promising. And one of the things that's also of interest to note here from the multi-source exchangeability model is these effective sample sizes, which with our information sharing are much higher than the actual sample sizes due to the smoothing across these exchangeable baskets. So for example, you can see, whereas the ovarian basket only actually enrolled 9 patients, it has an effective sample size of 73. And so this is a really powerful approach to basket study design. So that concludes kind of the technical-ish information that I wanted to present, and just some concluding thoughts here are that we can learn a lot from some of these case studies, but not everything. So there are a lot of open questions still in terms of, what went into the planning of some of these trials, what are some of the areas for improvement, what, if anything, could have been foreseen in the planning stages versus these, kind of looking back and being able to see exactly what went wrong, and what, if any, were the uncontrollable factors, and what exactly can we do better next time. So I do think there is a lot to learn here with the, keeping it very clear that I'm not criticizing any of the designs that were used in these cases when that was kind of what everyone was doing, and we didn't know what the results were going to look like at the time. But thinking through how we can avoid some of these pitfalls going forward in similar clinical contexts. So there are a lot of other areas for methodologic improvement in oncology trials, and a lot of issues that I didn't touch on, including the one that Nabiha brought up about late onset. You brought up late onset responses, but I think late onset toxicities are a big problem in the context of immunotherapy trials. And I didn't talk about toxicity monitoring at all, though there are a lot of new methods out there to monitor for both futility and toxicity in a joint way, which I think are really interesting to think about. Methods for non-binary endpoints are important. I think these binary response endpoints maybe are not always the best endpoints to choose for these trials anymore. Also, dose ranging studies, really trying to identify the ideal dose in the context of drugs where there is no maximum tolerated dose. I didn't even get into that in this case study, but as I had mentioned in the beginning, the maximum tolerated dose was never reached. And then these dose expansion cohorts looked at multiple doses, but it actually never really became clear for most of them which of the doses was the ideal dose. And this is just naming a few areas that are of interest to me, at least in the future. So I'll just leave you with this, which is if you know anyone or you are someone who's interested in this research area, I'm trying to hire a postdoc right now. So please check out my ad or send me an email to ask for details or apply, and some contact information here. I'm always happy to hear from anyone if you have ideas or questions after the fact. So please don't hesitate to contact me. And thank you so much for listening. Okay, I see a question here that says, in the basket design, does it require response to have the same definition, or could they be cancer specific? That's a really good question. I think the design has not been studied well to know what the impact is of, this isn't exactly in line with your question, but I am going to come back to this as well. One of my questions is about, do we have to have the same, null response rate in each of the baskets, or can we have different null response rates across the different cancer types, which I think is really important to your question about cancer specific, is that in a lot of these basket trials, we know that, there are already good treatments for some of these cancers and not so many good treatments for others. And I think for both of those questions, the answer is they can be cancer specific. I don't think the definition of response has to be exactly the same for each cancer. And I don't think the null response rate has to be the same for each cancer either, which is one of the benefits of that design. So that's a really good question.
Thanks. We have someone in the audience who's raised their hand.
Okay.
Erica, can you unmute them?
Emily, loved the talk. I had a couple of questions. One was, could you go back to the slide where you have GBM, and I just wanted to understand the meaning of all comers. Does all comers really signify that there is no inclusion, like there is no biomarker testing you're performing for inclusion?
Yeah, so I didn't look into some of these other cancer types as much as the bladder cancer, but yes. So I think what this schema is showing us is that, so for example, in the basket I focused on, urothelial bladder cancer, they enrolled all patients, but they had a specific interest in the PD-L1 targeted subgroup, that IC23 subgroup. But in glioblastoma, they did not look at PD-L1 at all. Same with melanoma here. And I don't know the biological reason for that or the design reason for that. So I can't speak to it, but that is what that means.
Okay. And can you go back to the second but last slide? Yeah, this one actually.
This one?
The one which had, the posterior probability threshold of 0.95.
Oh, this one? Yeah.
Yeah. So I was just curious if it's 0.95, if you look at the bottom two true rows, uterine and RCC, renal, so they actually have less than 0.95. So why are they true?
Oh, because it has to do with the ordering of the basket. So it's not actually just whether the posterior threshold is above that. It's so if you go back here, you see that it's that threshold multiplied by this ordering of the basket over the total number of baskets. So it's not strictly whether it's greater than that threshold. This is that false discovery rate control gives you a little leeway with that. But I should have mentioned that this was just an example using 0.95. You would probably actually want to calibrate this threshold to target a certain type 1 error rate and power of your design, which I didn't do in this case. I just was looking at a reanalysis and using an example threshold. So probably you would want to have a more strict threshold than the 0.95. It's a good question.
And finally, the network graph you showed us,
Yeah.
I was just curious, what did you use some open source software to draw this?
This is from the basket package that does the multi-source exchangeability models. This is one of the graphics you can get from that package, which I think I have here there's our journal citation for the paper about the software associated with this methodology, this Kane et al. Our journal paper.
Cool. Thank you so much. Really enjoyed the talk.
Yeah, thank you.
May I ask a question? I'm Hajime. Thank you for very nice talk. So I have a question about slide 21, your first part of the talk. So yeah, first question I had is the right-hand side, right? So you have the constraint on type 1 error and the power here.
Yeah.
So but is there any range of type 1 error and the power here? So
in this actual plot, this is just fake data, so this isn't a real example, but let me go to like the results I was showing. In this plot, I filtered these according to, I'm trying to remember, I think a type 1 error less than 0.1 and a power of at least 0.7, I think were the criteria I used.
see. Okay.
Yeah, so any designs that fell outside of that, I said we wouldn't be interested in considering them for optimization based on efficiency. Yeah.
I see. Okay, thank you. So another question is the choice of prior distribution. So I think you used the uniform prior beta 0.5, 0.5.
Yep.
So if we use some other prior, so the picture in slide 21 change a lot, or it doesn't change much?
It does not. So I did look at that. I looked at the sensitivity to the prior, and if you use a very informative prior, it will change. But if you keep I looked at a variety of like also other uninformative types of priors, some different beta distributions and other things, and it's not very sensitive to the choice of the prior.
Okay. Yeah. Thank you very much.
Yeah, you're welcome. Thank you.
Last call for questions. You can type them in the Q&A box, or you can put your hand up and we can unmute you. Okay. Well, thank you so much, Emily. That was really great. And that was very interesting. I think we all learned a lot, and we'll try out your packages. That's on my to-do list.
Thank you so much for inviting me. I really was happy to be here.
Okay. Take care, everyone. Thanks for attending.
Thanks.